Hello, and welcome to our lecture on prokaryotes. We'll be talking about how to characterize and classify them today. And so we'll be able to compare and contrast prokaryotes at the end of this lecture, known to cause disease in various species, with specific details from this PowerPoint, um, aka the model species. So we'll go over gram-positive bacteria, we'll go over some archaea, and then we'll go over gram-negative bacteria and how they are described. Finally, we'll be able to classify organisms as prokaryotes and eukaryotes depending on their cell structures. So we'll see how they divide as kind of a review from our cell lecture. And we'll also see a few other things, but I tried to keep it short and sweet, so let's get into it. So most prokaryotes have these general characteristics. Obviously, they're the most diverse groups of microbes. We can have so many distinctions of them. They have their whole side of the tree, and then they even have the archaea, if you remember that phylogenetic tree. Okay, so they're diverse. They thrive in all habitats, archaea being the very good examples of that. They are known to live in the deserts, in extreme sulfur vents and heat, and also in the Great Salt Lake. Remember, less than 1%, less than 1% of bacteria actually cause disease. Okay, so most of them can be utilized to have some form of benefit rather than actually causing disease. Okay, so now that's not to say most bacteria can be used, just most can be used, more can be used as a benefit than are pathogens. Okay, and then as you know, they exist in a variety of shapes and sizes. Even though their scale is the same, they'll still have different sizes within that scale, and also their shapes, bacillus and coccus, being the two main examples that we need to know. So with that, there's our coccus. You should already know that that's a sphere. Here's our bacillus. That is a rod. All right. We have some coccobacillus. I would not worry about that one too much. Vibrio, you definitely need to know because there is a disease called Vibrio cholera or cholera, and that is a very relevant disease right now. After Haiti, the cholera tends to come after misfortunes in low impoverished areas. We have spirillum and spirochete you're going to want to know, and then of course a few others that we see here called pleomorphic and star, but those are not as important as coccus, bacillus, vibrio, spirillum, and spirochete. You're going to want to know those shapes, especially for the first test. All right, and we've mentioned these before, but endospores are extremely common in gram-positive bacteria known as bacillus and clostridium. In fact, Bacillus cereus, or B. cereus, is what we used in the lab just the other day to see a gram-positive example. They use these spores as a defensive strategy against unfavorable conditions. When the conditions are going to kill them, they don't have any water, they don't have any sugar, or there's a toxin or an antibiotic, they will go under sporulation and make a spore. Okay, these are a concern to healthcare professionals, governments, food processors. These are the number one concern because they are so hard to get rid of. Okay, they are hard to get rid of. Okay. and they are survival structures. They are not reproductive structures. Okay, the term spores kind of means that sometimes, but these are survival structures. All right, and for binary fission is how they divide and replicate. When you get a chain of bacteria that kind of line up in a line, this is why. When they bud off, they kind of stick together and form that chain, because then these two will divide and make a bigger chain. Okay, but binary fission requires the DNA inside the cell to replicate first. Okay, it must replicate, duplicate itself, and then it will divide those apart, and then it will start to put proteins here. They're called the FTSZ proteins, and they actually start to close and pinch that gap, and now you have two what we call daughter cells. Okay, and they can separate if they're individual bacteria, or they may stay in their chain, or they may even cluster by changing their plane of orientation of where they're dividing. Okay, so binary fission. Okay, binary meaning into two parts, and fission meaning cut in half. All right, and just so we can have another example, there's another way they can reproduce. It's budding. All right, but again, one of the main ideas here is that first the DNA needs to replicate in order to have two cells. You can't have a cell without DNA, and so this is how we get another form of reproduction in bacteria is by budding. Okay, and then this daughter cell will grow before it decides to bud off and make more. Okay. As I was mentioning, we can have different forms of cocci and bacillus, and it is all relevant, all according to their plane of division. Their plane of division will tell you which kind of bacteria you get. So if you have two cocci, you are a diplococci. If you have a chain of spheres, you're going to be strepto. And if you have them in a quad or a four pair, it'll be called a tetrad. And then if they are branching and dividing along any plane, they are staphylococci. So branch equals staph. Okay. So remember that as we head into our bacillus. They also have that diplobacilli distinction, and they have a streptobacilli distinction. Okay, so a chain and a pair. Pair and chain. Okay. And finally, we will end this video with the modern prokaryotic classification. Remember I said we kind of have them branching off everywhere? Well, we use gram-negative bacteria and we use gram-positive bacteria to sort these guys. Okay, and if you can see here, this tree of life is the bird's eye view of this tree right here. So we're looking down at the circles right here. Okay, right here is looking down at the circles. All right, and in it we can see that we have three domains. Archaea are very separate from the rest of them. Archaea will be described next. And you can see the gram-positive and gram-negative bacteria are on their own branches, but are very related as well, okay, because they are kind of close. And deeply branching bacteria are more related to gram-negative bacteria, is what this would imply as well. All right, and these are all based on the relatedness of ribosomal RNA, so ribosomal RNA sequences. You take those and line them up, and you see how they differentiate over time, and you can come up with this tree. Alright, so we'll get into archaea and the other gram-positive and gram-negative bacteria next.